Hello everyone, welcome to week 12 of our Biochem Weekly Reviews. We are very close to the end of the semester, as I'm sure you can tell by having just taken your third exam. And then obviously you just have your last exam, and this is, one of the last 3 weeks of the semester in terms of content. We are very close. And actually, I would say we're probably to the point where, you guys know exactly what's coming up next. This week we're talking about glycogen and the pentose phosphate pathway. Next week will be the the Krebs cycle, the citric acid cycle, tricarboxylic acid cycle, etc. There's about 1 million names for it. And then the last week will be the electron transport chain. And so we're very near the end. And so yeah, we're at the end of the semester. We're just going to jump right into the content this week. This is, admittedly, I'm not the biggest fan of the glycogen content, but the pentose phosphate pathway I think is so cool, so interesting. And so this is one of the weekly reviews that I'm really excited to be giving. So I'm just going to go ahead and get started. So the first question you guys see on your screen is, why doesn't glycogen phosphorylase use ATP to cleave glycogen? So I'm going to approach this question the way I've been approaching everything. Let's think about energy. Let's think about, the big picture. So glycogen phosphorylase is used in glycogenolysis to break down glycogen into glucose 1-phosphate, which can then be converted into glucose 6-phosphate for us to use as energy when we don't have enough glucose in the blood. Okay. So in other words, we use glycogenolysis when we need energy. Okay. So if it were to use ATP, it would waste energy, okay? And generally speaking, since we're glycogen, and I believe we've talked about this very briefly, but glycogen is effectively glucose linked to glucose, linked to glucose, linked to glucose, and so on. And then somehow you introduce a phosphate group, and you cleave, switching colors, you cleave from the left side going to the right, and you make this a G1P, and then you have glucose to glucose to glucose, and so on. And these are all alpha 1,4 linkages. Okay, so that's great. But what does that have to do with, how does that fit into energy? So we use this enzyme when we need energy. So it doesn't make sense for us to use energy when we need energy, right? The two don't match. And so that's why I can tell you all already, B is going to be wrong. If it used GTP instead of ATP, it would be the exact same thing because the energy is almost identical. Okay, so that's out. ADP, while yes, it does have phosphate groups because it's a diphosphate, ADP is generally a sign of low energy status, and so it's also out. And then finally, the reaction itself doesn't require that much energy because, interestingly, it uses inorganic phosphate. Okay, so this prevents need for ATP, allowing us to preserve energy. Okay, all right. So what happens to glycogen phosphorylation with a high concentration of ATP? So the word that I just said, glycogen phosphorylation, is the same as the glycogen cleavage reaction of glycogen phosphorylase. Okay, it's a little bit of a weird reaction. So normal reactions that we've been using are hydrolysis, right? Where two things bind and water gets kicked out, like with amino acids turning into peptide bonds, right? However, this is called phosphorylasis, excuse me. And so glycogen phosphorylation is the same as glycogen cleavage, just to make sure that's clear to everyone, okay? So again, let's think about what ATP is, is with respect to the glycogen phosphorylation and to the glycogenolysis reaction. So again, just like with number 1, we use glycogen phosphorylation when we need energy, okay? So now I'm thinking, need energy, but if we have a high concentration of ATP, we have energy. right? So that's telling me that somehow we're going to be inhibiting the glycogen phosphorylation reaction. Now you guys see that there are four options that look very similar. Or I should say, I'm sorry, four options, two of which look very similar. So I can already knock these two out. But what's the difference between B and D? Okay. So there's not really a difference. Well, let me reword that. Generally speaking, there isn't a difference. However, it's important to take note. This is saying the pathway in general is inhibited. This is saying a specific enzyme is inhibited. So while both are correct in this case, it's important to note, so for example, if I said PFK2 activates glycolysis, or more specifically, fructose 2,6-bisphosphate, which we learned last week, if I said that's a glycolysis activator, that's true. If I said that's an activator for the PFK1 reaction, that's also true. But if I said that it's an activator for the phosphofructoisomerase reaction, or phosphoglucose reaction, excuse me, that's obviously incorrect. And so I'm more including this question just to say, be careful about wording. There are plenty of ways to trick you. Don't worry, they love to make you miserable. And the graphs, I should say, these that you see here, are kind of representative of that. So important to note, cooperativity is shown here. How can we tell? There's that sigmoidal curve. It starts low and then it increases, okay? So just a recap of sigmoidal curves, the first bit right here is basically the amount of effort, concentration. Basically, this is the first binding, okay? But once the first thing has bound, you see a drastic increase in the binding of everything else and in the rate of this reaction, okay? In other words, the binding of inorganic phosphate starts off slow, gets faster, okay? All right, ATP is a negative effector or an inhibitor. Inhibitor of glycogen phosphorylase, and glucose 6-phosphate has a similar effect, okay? So an inhibitor decreases the affinity of the enzyme for inorganic phosphate. Let's look at the graph and make sure that makes sense because it has been a little while since the enzyme unit. Okay, so we see that the Vmaxes are roughly the same, and actually if this graph kept going, it would end up leveling off to be pretty much exactly the same. The difference is how much longer this ATP curve takes to reach each point. So if we think about Km, and I know you guys are probably tired of hearing about Km, but if we approximate, Km for the regular curve would be here. Km for the curve with the inhibitor would be here, plus inhibitor. And so since Km is a measure of affinity and specifically has an inverse relationship with affinity, what it's saying is that with an inhibitor, we have a lower affinity for inorganic phosphate. Okay. All right. So finally, part C of this graph, AMP. So AMP and ATP show very similar reciprocal regulation effects, like we've talked about before. When we talked about glycolysis, AMP was kind of a signal that we don't have a lot of energy. We need to do more glycolysis to get that energy. ATP was the opposite. We have plenty of energy. Shut down glycolysis because we're just wasting the glucose, right? This is the same here. When you have a lot of ATP, you don't need glycogenesis. I'm sorry, you don't need glycogenolysis. You need glycogenesis, excuse me. So when you have a lot of ATP, when you have high energy status, you do not need to break down your glycogen into glucose to make more energy, okay? When you have ATP, glycogen phosphorylase is inhibited. When you have AMP, AMP tells you that you have not enough energy, effectively, and that tells you make more. How do you make more? When you have a low energy status, glycogen breakdown is stimulated, and this reaction happens faster, and more of it happens, okay? All right, so the next question is, what kind of modification converts glycogen phosphorylase between its A and its B form? Okay, so this is a weird topic because we haven't talked about specific enzyme kind of modification in this amount of detail yet, but this is fairly regular for the body, as I'm sure you'll learn if you've taken, or if you're going to take Biochem 2, or if you've taken a few bio classes. But generally speaking, so specifically, or I should say, the glycogen phosphorylase has, as it says, an A form and a B form, and we're going to talk more about both of them in the next question, so I'm not going to go into much depth about it. But the key here is that the conformational change, induced by the conversion between A and B makes one of them persistently active and one of them sensitive to allosteric regulation and therefore less active. And the only difference between them is phosphorylation. Phosphorylation. Okay, so let's say the A form, and we're not going to talk about which form is which yet. If the A form is the active form, then we phosphorylate to get to the A form. Or maybe we phosphorylate to inactivate to the B form. But effectively, we're phosphorylating to convert between A and B, and therefore we're dealing with covalent regulation. Okay, so just a quick recap, allosteric regulation is non-covalent bonding. Okay, so it's like what we see up here. These are all allosteric inhibitors because they bind at a site that's not the active site, and they don't actually form covalent bonds. Okay, what they do is they fit into the enzyme using either the lock and key or the induced fit hypotheses, or very similar since it's not the active site, but something along those lines. Okay. We do not cleave the phosphorylase, so we can use cleavage with the example of zymogens that we talked about. That must have been week 6 or 7 maybe. And while cooperativity is a factor, it does not actually modify the enzyme, right? So we talked about cooperativity up here. It does play a role in the enzymes, in the regulation of the enzyme, but that is not the modification we're talking about here. Okay, so I'm going to zoom out so we can talk about glycogen phosphorylase. Okay, so, okay, that's perfect. All right, so I'm going to start off by saying we're looking at the MWC model. Okay, so with the MWC model, remember, just a quick recap, you have the tense state and the relaxed state, and they're in equilibrium with one another. The tense state is inactive, or I should say relatively inactive. The relaxed state is relatively active. Okay, so that's what you guys see along these these vertical equilibria, is the non-covalent control based on activity, based on concentrations. Okay, so looking specifically at, so that's down here. Let's look specifically at the covalent control, which the highlighting is barely visible. Let's look at covalent control. Okay, so using phosphorylase kinase, we can add a phosphate group to the phosphorylase, or really 2 phosphate groups, and make phosphorylase A. Okay, using phosphoprotein phosphatase, which is a mouthful, or specifically phosphoprotein phosphatase 1, we can remove the phosphate group to make the inactive B form. Okay, and when I say inactive, the reality is the relatively inactive. Okay, and I know that they both say inactive right here, but that's because we're dealing with the T state. However, in reality, A is, like I mentioned, the persistently active one. Persistently active. And B is sensitive to regulation. Okay, and we're going to talk about how that makes a difference when we get to the MWC part. But first, I just want to make sure we know the difference. Okay, so then let's look at the MWC part. So both of them are currently in their tense form. Okay, however, they can be activated or inhibited in some way or another. Both of them are affected by glucose and caffeine. Okay, and we're going to talk about the other parts later, but for now, glucose and caffeine. And let's think about what glucose and caffeine are. Okay, well, glucose specifically first. Glucose tells you you have glucose in the blood. If you have glucose in the bloodstream, you don't need to convert your glycogen to glucose, right? Therefore, glucose converts your A form, or I'm sorry, your active form, your R state, into the tense state because it turns off the phosphorylase. Okay, caffeine. Caffeine is fascinating because everyone knows you drink coffee in the morning to wake yourself up. You, whatever the case may be. However, you don't, caffeine doesn't actually wake you, or it does wake you up. I don't want to say it doesn't. Let me reword that. Caffeine is not a long-term solution for this reason right here. While coffee does wake you up right away, it leads to these kinds of like quote unquote crashes because it eventually converts your phosphorylases into their inactive states. Okay. All right. So now let's talk about the elephant in the room: AMP, ATP, glucose 6-phosphate. And what I mean specifically by that is, why are they on one side and not the other? Okay. And this comes down to what I was saying earlier about this being persistently active and this being sensitive to regulation. Okay, so when you have, and I'm going to focus on the left, I'm going to be talking about the B form, but I'm specifically going to be looking at the right side of the equilibrium of the B form. So I'm looking at the ATP and the glucose 6-phosphate. We'll talk about AMP after. When you have ATP or glucose 6-phosphate, well, glucose 6-phosphate, for the exact same reason as glucose, is telling you this is something that can be converted to energy. ATP is energy. Let me reword. ATP has high energy bonds that we can use. I keep saying it is energy. That's not entirely accurate, right? So ATP provides energy. Glucose 6-phosphate provides ATP. Therefore, they can inhibit phosphorylase B because when we have these in our bloodstream, we don't need more phosphorylation, or we don't need more glycogen breakdown. AMP has the exact opposite logic, just like before. When you have AMP, that's telling you have low energy status. Low energy status tells you, let's get more energy. How do we get more energy? We release glucose 6-phosphate from glycogen. Okay. Why aren't they present in A? Because A is persistently active. Because A doesn't want to be inhibited or regulated by the presence of glucose 6-phosphate or ATP. Okay. So the other thing that isn't really shown as clearly is the actual equilibriums. I've been just kind of saying they're in equilibrium with the MWC model, but we haven't given you any actual information about these equilibriums other than that they're affected. So specifically looking at the purple parts now, so the lines only. That's all we care about is these lines. If we remember, the equilibrium between the tense and the relaxed form is L, and it's T0 over R0. Okay. Well, L of B is significantly greater than L of A. In other words, the tense form of B, or the ratio of tense to relaxed, is greater than the ratio of tense to relaxed, and I just repeated that. For the B form, it favors its tense state, while relative to the B form, the A form has more relaxed state. Okay, in other words, this equilibrium favors this equilibrium favors this. So when we say that the A form is persistently active, we mean partly that it's not being affected by regulators, and partly that it's just more active because of that, because of the way the MWC model, or because of the way that the equilibrium is with respect to the MWC model. It's probably a better way to phrase that. Okay. All right. So, any questions about that topic? I know it's a bit weird. Okay, then let's jump into the next part. What makes phosphorylase A more active? Spoiled that for myself. It's okay, it's important to write it as well. So first, less regulation. And second, the L of A being less than the L of B per MWC. Okay? All right, excuse me. So that's phosphorylase, or that's glycogen phosphorylase. However, I told you guys phosphorylase kinase and phosphoprotein phosphatase, I told you they existed and moved on from them. The reality is it gets more complicated, and I'm going to be zooming in more so we can look at this. Okay, so this is what's called a signaling cascade. Okay, and a lot of you guys may have heard about them if you took a biology class, but if you haven't, that's okay. We're going to go over them. So cascade regulation or signaling cascades or whatever you want to call it are effectively thing A, and I'm going to keep this as vague as possible, causes something to change in thing B, which causes something to change in thing C, which causes, and so on, right? And I keep this very vague because signaling cascades can activate or inhibit or both. And so, for example, you may see that some hormone causes something to inactivate, which allows something else to activate or whatever. And so it gets a lot more complicated. And so I'm trying to keep it vague for now, but let's look specifically at the signaling cascade for, eventually, phosphorylase kinase and glycogen phosphorylase. Okay, so we're going to start with the hormone. Okay, so a hormone is almost always the start of these signaling cascades because hormones affect biological conditions, right? So the hormone in question here is either adrenaline or glucagon. Okay, we're going to talk about each of them real quick. So glucagon, remember, is kind of the opposite of insulin for convenience. When you have low blood sugar, like we talked about last week, and I'll flip to that real quick. So I showed you all, was it not last week? It might have been the week before. I apologize. Probably the week before. Yeah, this is, okay, never mind. I was trying to find this and I missed it. So low blood sugar causes glucagon to be released. There we go. Okay, back to here. So glucagon is made in response to low blood sugar and stimulates the mobilization of glycogen. Okay, so glucagon is just kind of for your body's natural homeostasis. Epinephrine, adrenaline, you guys have heard of it before, of course, it triggers the fight or flight response. And so we need energy in order to have that response. So either of these enzymes will cause glycogen to be converted to glucose, or specifically glucose 6-phosphate, in order to give the body more energy. Whether that energy is needed for maintaining the health by keeping our blood sugar at a reasonable level, or so that we can move around, we need more energy, right? So how does it do this? First, it activates adenylyl cyclase, okay? So I want to very briefly mention these two words. Adenylyl comes from the same word as adenine, which is one of the nucleotide bases, and specifically adenosine, which is the first letter of adenosine triphosphate, or ATP. Phosphate, or ATP. Okay, so cyclase we'll talk about in one moment. Okay, so what does the adenylyl cyclase do? It converts ATP to what's called CAMP. Okay, so AMP we know, it's adenosine monophosphate. The C out front it stands for cyclic. Okay, so this is cyclic AMP, and that's what the cyclase does. It causes this to become cyclic AMP. Okay, all right. Now what does cyclic AMP do? So there are something dependent protein kinases, and I say something dependent because there's very similar signaling cascades with a whole host of other hormones and a host of other eventually like downstream enzymes. And so there are protein kinases that are activated or inactivated because of the presence of something. Cyclic AMP, or as I'm going to start calling it because that's too long a name, CAMP, is one of those molecules that can trigger a protein kinase. And specifically, it triggers CAMP-dependent protein kinase. Okay, and it activates it, right? So active, and I'm just going to go ahead and highlight the last two since I know you guys can see them. It's always activating. Okay, so when you now have a protein kinase, this protein kinase can do what kinases do. It can phosphorylate something, but it doesn't phosphorylate glycogen phosphorylase. And this is something that I think is really complicated, or not complicated, confusing, or kind of unintuitive to a lot of people, is you phosphorylate the thing that eventually phosphorylates what we're looking for. So in other words, phosphorylase kinase is the enzyme that I told you triggers the conversion of B to A, of phosphorylase to B to phosphorylase A. Well, that's also regulated by phosphorylation. So as you guys can see here, ATP to ADP, phosphorylase kinase to phosphorylase kinase with a phosphate group. That's that's the enzyme, or I should say, that's the reaction catalyzed by the protein, or the CAMP-dependent protein kinase. Okay, and that's another thing, is keep track of this name, protein kinase. Okay, so it's not a glycogen kinase, it's not a phosphorylase kinase, it's not phosphoprotein kinase, it's a protein kinase. And so exactly as it sounds, it phosphorylates the protein, okay? And specifically this protein, all right? So now, since it's phosphorylated, it's in its active form and can finally activate glycogen phosphorylase by converting it to glycogen phosphorylase A. One thing to take note of is this uses 2 as opposed to 1 ATP because, as you guys see in this figure right here, we phosphorylate it twice. Okay? All right. So now let's look at the other words on this slide. The cascade amplifies the hormonal signal. Okay? So the easiest way that I think of this is if I tell 2 people something, and then each of those 2 people tell 2 people something, and then each of those 2 people tell 2 people, and so on, right? That's what's happening with this hormone. This hormone will activate 5 adenylyl cyclases. Each of those 5 adenylyl cyclases will convert 5 ATP to 5 cyclic AMP. Each of those 5 will, and so on, and so on, and so on. And so you get a what's called a signaling cascade, or, I'm sorry, a amplification of the signal because it's activating more and more and more as you go down each step. Okay? All right. So we've talked about glucagon. Now let's talk about glucagon, quote unquote, regulation, right? So glucagon secretion is inhibited by insulin, which makes sense because the 2 are opposite. Okay? So this is really interesting. In diabetics who either can't produce insulin or whose body doesn't react to insulin, glycogen will still be degraded even when glucose is high because this signal, this inhibition isn't happening, okay? Therefore, we're never being told to stop, okay? Glucagon keeps being secreted, and since glucagon keeps being secreted, glycogen keeps getting degraded, okay? Since we're not told to stop, we keep doing it, right? Okay, any questions about the signaling cascade involved with glycogen? Okay. Good, because we haven't gotten to that part yet. Cyclic AMP. Okay. So like I mentioned, so actually I didn't mention this, so there are a couple of other quick terms that I'm going to mention. So, excuse me, I don't know why that's, okay, there we go. Okay. So, the hormone, or let's just say glucagon, is called the primary messenger. Okay? The body detects something is wrong, a primary messenger is sent out to address that. Well, the hormone has to activate something, and so one of these intermediates, or really most of these intermediates, specifically cyclic AMP, what we're going to focus on, is called the secondary messenger. Secondary messenger, because it's after the primary, right? And so, yeah. So that is cyclic AMP kind of in a nutshell, or I'm sorry, that's regulation in a nutshell, but we still have to talk about cyclic AMP. All right? So cyclic AMP is a secondary messenger, or second messenger, because it's an intracellular agent that moves around. Okay, so the enzyme adenylyl cyclase is membrane associated. It is attached to the phospholipid bilayer. Cyclic AMP is not. Cyclic AMP moves around. It's an intracellular thing that's floating around the cytosol. Okay, but cyclic AMP is complicated. All right, and here's why I say it's complicated. So CAMP-dependent protein kinase, which I'm just going to call the kinase, is regulated in the sense that it has 4 subunits. And once again, we're going to use colors for these. So we have 2 of the first subunit and 2 of the second subunit. Okay, and the reason I specifically use red is the 2 at the bottom are called regulatory subunits. Okay, the 2 at the top are called catalytic, the 2 at the bottom are called regulatory. Catalytic, regulatory. And their names are pretty spot on in this case. Catalytic stuff wants to catalyze reactions. Regulatory, there we go. Regulatory want to regulate that. And so effectively what happens is by binding CAMP, CAMP, and so on, these subunits dissociate from one another, leaving the catalytic subunits free to react to catalyze, and so it creates the active kinase. Okay. All right. So we've talked about glycogen breakdown. Now let's talk about glycogen synthesis. So glycogen synthesis is not fun. I told you all I'm not a big fan of it. However, I do like this figure. I think these kinds of figures are great, and so we're going to be talking about this figure, or we're going to talk about it with respect to this figure. Okay. So glycogen synthesis follows this pathway, right? These, or I should say, these are the reactions of the glycogen synthesis pathway. This is the to the left is the regulation stuff, and then the degradation is the same in the sense that this is the degradation pathway, and the things to the right are the things that affect it. Okay, all right. So let's talk glycogen synthesis. Glucose is activated for synthesis. So when I say activated for synthesis, what I mean is glucose plus ATP gives us glucose 6-phosphate. That's fine. You can isomerize glucose 6-phosphate to glucose 1-phosphate. Take some energy, but it's not the worst thing in the world. And so you can go backwards, right? But once you get to UDP glucose, so UDP is uridine diphosphate, and you form UDP glucose, that is the point which we've activated it, so we've given it the energy to make glycogen for us. Okay. So interestingly, what's the word I'm looking for? So in the same way that we use ATP hydrolysis, we couple reactions to ATP hydrolysis. We do something very similar with the UDP. So glucose 1-phosphate plus UTP gives us UDP plus inorganic phosphate. We couple that with, I guess I'm going to be over here to the right side, inorganic phosphate plus H2O, or I should say, I'm sorry, pyrophosphate, not inorganic phosphate. Pyrophosphate gives us 2 inorganic phosphate. Okay, so this cleavage right here catalyzes this reaction right here. Okay, because it takes energy. All right, so anyways, so let's talk about the regulation stuff. So very briefly, we're going to talk about the synthesis pathway, and we're going to talk about the degradation pathway, and then we're going to talk about in more depth. So glucose plus ATP gives us G6P, same as glycolysis. G6P is then isomerized to G1P. So I'm not sure if you all remember I don't know if I actually mentioned it now that I think about it, I apologize. That uses phosphoglucomutase. So we've used a mutase before. It's very similar to an isomerase. Phosphogluco, because it's a glucose with a phosphate. Makes sense. Okay. So then using UTP, you add UDP to glucose, and then using glycogen synthase, you create glycogen. And that's a very, very broad generalization. Like that's the quickest version of that I could give. Partly because we're going to go over it a little bit more later, partly because I don't like glycogen synthesis. But yes, so glycogen synthase gives us glycogen. Then, like I mentioned, glycogen through glycogen phosphorylase gives us glucose 1-phosphate, and we just do the reverse of this, right? Glucose 1-phosphate using that phosphoglucomutase gives us glucose 6-phosphate, which can then proceed through glycolysis. Okay, so you guys see low blood sugar on this side, and I love these graphs because I think they make sense. I don't like these graphs because they can confuse people. So when you have low blood sugar, you release glucagon. Let's talk about glucagon once again, or epinephrine if you need the fight or flight response. But let's talk about glucagon specifically. Glucagon, when you have low blood sugar, tells you need more sugar in the blood. You need more glucose, and therefore it inhibits glycogen synthase. Okay, so the things on the left and right, the far outsides, are the same because it inhibits one. And then if you notice, the exact same things on this side, except now it's activating. Okay, so once again, I draw you to this right here. Reciprocal regulation is the key here. I'm going to actually put it in bold now. Because of the fact that the thing that activates one inhibits the other. Okay, that's so important, not just in this, but in most reactions, because that helps us be more efficient. Okay, and just rolling through the rest of this really quickly, glucose 6-phosphate. If you have high glucose 6-phosphate, you have enough energy. Therefore, you're activating glycogen synthase to store some of that energy, and you're inhibiting glycogen phosphorylase because you don't need more glucose 6-phosphate. ATP and AMP we've already covered, so I won't go over them in depth. Inorganic phosphate. Let's talk about this bit. So inorganic phosphate is interesting. It activates glycogen phosphorylase. In other words, it favors glycogenolysis. Why? Okay. The way that I think about this is not based on like a ton of logic. It's just the way that I like to remember it. And it's the fact that, there's my mouse, I apologize, ATP. When it's used, when it's hydrolyzed, you get ADP plus inorganic phosphate, right? So in the same way that ATP is it tells you have a lot of energy, well, inorganic phosphate tells us we've used a lot of energy, and therefore it favors glycogen phosphorylation because it's telling the body we've used a fair amount of energy, we might need to look at getting more. Okay, and that's kind of the logic behind inorganic phosphate. Okay. Right. So then, shifting gears a little bit, what is used to prolong ATP availability in the muscle? So this is very kind of random in my opinion. The thing about it is glycogen, the purpose of glycogenolysis is to provide you with more ATP. But at the end of the day, you're hoping that you eventually have enough, but you haven't gotten there yet, right? Glycogenolysis is a process. It takes some time, and then after you've made glucose from glycogen, you then have to release it all over the body, and it has to get there, right? And so you need something else in the meantime, and that something else is phosphocreatine. Okay, so phosphocreatine is, I mean, exactly what it sounds like. It's creatine with a phosphate group. Chemists are so great at naming things. But in all seriousness, so the point of phosphocreatine is it's a short-term source of ATP in the muscle, and it can phosphorylate ADP to make ATP. Okay, so looking at a graph of this, and I think I put the graph, yes I did. Okay, so the orange line at the very beginning, so this line right here, is basically saying for the first 5 seconds maybe, and that's being generous, of your exercise, you have ATP, and then you run out. Okay, so what do? You use phosphocreatine. Okay, so phosphocreatine will kind of bridge the gap between ATP and basically glucose and glycolysis and stuff. Okay, so you have a little bit of ATP floating around in the body, or in the muscles, I should say. The rest of your energy comes from phosphocreatine for a little bit, and then it switches over to anaerobic metabolism. Okay, so what is anaerobic metabolism? Remember that glycolysis is an anaerobic process. Okay, so glucose takes over, glycolysis takes over. Okay, and it lasts a little while, and then we run out of glucose, right? Or let me reword, we don't necessarily run out of glucose. It might be more appropriate to say we've used enough glucose and made it into pyruvate that now we can use the electron transport chain and the citric acid cycle. So TCA plus ETC is the electron transport chain. All right, but eventually that's going to run out too as we run out of oxygen, right? So anaerobic metabolism, or I guess I should say this is the process. This is where energy exists, basically, and how it exists in our muscles. Yeah, okay, so once again, time is a factor, okay? And the reason it comes down to time is glycolysis takes time to set up to actually give us ATP. Remember that glycolysis is 10 steps, and in the first 5, we use 2 ATP, right? And yes, obviously, we more than make up for that later on, but it tells us that our ATP, well, first of all, that's part of the reason we're depleting ATP, but second of all, that means that we're not getting the payoff for glycolysis until reaction 7, reaction 8. 7 to 8 is when we finally break even, and then actually, let me reword that. We actually get the payoff in reaction 10. It finally pays off once we finish the entire cycle. That's why it takes so long for us to get energy from ATP, or I'm sorry, from glycolysis. It takes time, and then eventually it predominates. And then eventually, as we start running out of glucose, as some of the pyruvate that's made is able to enter into the mitochondria and go through the ETC and the TCA, we get more aerobic metabolism, and then eventually that's going to run out too. And as that's running out, we shift over to lactic acid, and I guess glycolysis would kind of uptick as well from there. Okay, all right. So what I personally believe the boring part is now over. Now the pentose phosphate pathway. Okay, so before we start the PPP, I'm going to start off by explaining we've been talking NADH this entire chapter, this entire class. This is just very poorly written D. I'm going to introduce NADPH in this unit. NADPH is very similar to NADH in that it provides reducing potential for the body, but NADPH is specifically used for building up. It's for biosynthetic reactions like building muscle and so on. Okay, NADH we tend to use a lot more for energy. NADPH we use to build. Okay, so the pentose phosphate pathway is very interconnected to glucose, as you'll see in a little bit, and it provides reducing power and carbon compounds for biosynthetic reactions. Okay, it's got a couple of other names. We're not going to worry about it. We're just going to call it PPP. Okay, right. So the PPP is now split into two branches: the oxidative branch and the non-oxidative branch. Okay, and one thing that I find fascinating about this, and one of the reasons I think it's so interesting, is that you can do either, both, or neither, and everything stays pretty similar. Like you're not wasting what's it called, the glucose or any other molecules if you choose to not do the PPP, and you're getting stuff back even if you do use it. Okay, so let's go ahead and jump into the oxidative branch. All right. So I apologize, that carried over from my notes. Okay, so let's start off with what are we dealing with? And we're dealing with glucose 6-phosphate. That's what number 1 is. Glucose 6-phosphate. Hopefully writing that for the very last time eventually. Okay, all right. And what you guys see right off the bat is this is a reduction. Okay, I don't know the enzyme name. Maybe it's a dehydrogenase, maybe it's a reductase, maybe it's a whatever. But I know we're reducing something. And actually, the enzyme name is glucose 6-phosphate dehydrogenase. Glucose 6P dehydrogenase. Okay? And that creates the second structure, which is called phosphoglucanolactone. Excuse me, I don't like the long names because I always get them mixed up. Phosphoglucanolactone. Okay. All right. Now this is a weird structure, and the reason I say that is because you've effectively got this ester right here, and it's not entirely stable as an ester. So there is an enzyme catalyzing the next reaction, for the record, but you don't need it. Okay, and that's something that is really fascinating, is this next reaction using glucanolactonase. Glucanolactonase. I apologize. I encourage you all to read this from your slides rather than my handwriting. I'm here to just give you an overview. But effectively, this is relatively unstable. Unstable. And so this will happen pretty much spontaneously, and it'll happen with an enzyme catalyzing it, but it gives us this, which is just phosphoglucanate. Okay. Phosphogluconate. And I should be specific, it's 6-phosphoglucanate and 6-phosphoglucolactone. But yeah, okay. So this is much more stable. And that allows for the next reaction to happen. The next reaction is, once again, a reduction. okay? And since it's a reduction, you're using another dehydrogenase, and in this case, it's, I don't know if you guys can guess it, but it's 6-phosphoglucanate dehydrogenase. Phosphoglucanate dehydrogenase, okay? All right, so interestingly here, we lose CO2. So this is something that I encourage you to do really when we get into the citric acid cycle, but it's especially relevant here. This is 6 carbons. This is 6 carbons, 6 carbons, 5 carbons, okay? It doesn't make a huge difference, but you guys will see why we do that in a minute. Anyways, the reactant is called, really, there's 2 steps kind of mixed into here. We don't care about both of them. We just get to ribulose 5-phosphate. Okay, so ribulose 5-phosphate. Okay. So let's pause and ask ourselves, why is this called the oxidative branch, and what can we expect from the next one? So it's called the oxidative branch because we are oxidizing glucose. I mean, that's really what it comes down to. We're using the reduction, or the NADP plus is getting reduced in order to oxidize glucose, hence oxidative branch. Okay. Next, it's called the pentose phosphate pathway, right? And ribulose is a large part of the reason why. Ribulose, if you guys weren't sure, based on this figure right here, is a pentose. It's got 5 carbons, right? And it's got a phosphate group. Okay. So in other words, it converts a 6-carbon sugar to a 5-carbon sugar in the oxidative. Oh, I scrolled way too far. Okay, next, the non-oxidative branch. And the non-oxidative branch is fascinating. I think this is one of the coolest things. So from here on, I'm going to say we have 2 ribulose 5-phosphates, okay? And the reason I say there's 2 is because they can do one of 2 things, okay? And you guys can see that split thing going on right there. We're going to talk about each of them. So from your ribulose 5-phosphate, you can either get ribose 5-phosphate or zolulose 5-phosphate. Okay, so let's talk about the first reaction, or I should say the top reaction. And the top reaction is an isomerization, excuse me, which effectively moves the carbonyl functional group up 1 carbon. All right, so that's an isomerization. Reaction 5, I guess I should kind of number these for you guys, shouldn't I? Anyways, the other reaction is called an epimerization. Okay, so let me write that out. Epimerization. And this epimerization changes the functional groups on a single, attached to the same carbon. So while the isomerization moved a functional group from 1 carbon to another, this moves a functional group on the same carbon. And specifically, we're looking at carbon number 3. The OH group was to the right, the hydrogen was to the left. Down here, it's the opposite. Okay? Not the most important thing, but it's just interesting to know the difference. I believe you could be asked a question about isomerization versus epimerization, which is why I mentioned it, though. Right? So once again, we're still at 5 carbons, but now the reason that I said we have 2 of these comes into play. Because the next reactions happen in pairs, not in ribose is converted to ribulose is converted to whatever, okay? So the next reaction, which is reaction 6, is zolulose 5-phosphate plus ribose 5-phosphate, which add up to 10 carbons, undergo a reaction catalyzed by a transketolase. Transketolase. And if you guys couldn't tell, it transfers a ketone. Or I should say it transfers at a ketone, because what it does is it effectively transfers 2 of the carbons from zolulose to ribose. Okay, so we're chopping right here, and these all get attached, I believe, at the very top, if I'm not mistaken. So yeah, so if I'm not mistaken, and it has been a little while since I've seen the specific mechanism, but I believe these 2 are the same carbon. Okay. All right. So that's a transketolase, and once again, we're looking at 2 sugars. One is 7 carbons, one is 3. So the 7 carbon is called cetoheptulose 7-phosphate, and the 3 is called glycerin aldehyde 3-phosphate. Okay, all right. So now the next reaction is very similar, except instead of a transketolase, it's a transaldolase. Transaldolase. Okay, exactly as its name suggests, it transfers at the aldolase. And so this is the aldolase that we're looking at right here, but it's not going to move it over to our heptulose. What it's going to do is it's going to transfer a carbon to heptulose. And again, I believe, if I'm not mistaken, that it's this carbon that gets transferred onto here to create this, okay? So again, same number of carbons, keep that in mind. Carbons, we have 4 and 6 now because we have erythrose 4-phosphate and fructose 6-phosphate, okay? All right, so now this is where it gets a little bit weirder. Fructose 6-phosphate, we are happy with. We could stop here if we wanted to. Also, I just realized I forgot to mention this when I got here. Glycerinaldehyde 3-phosphate is glyceraldehyde 3-phosphate. I forgot this is an old image, and I completely forgot to mention it. Glyceraldehyde. I apologize for that. So it's G3P. Okay. So this is what I think is the most fascinating part of the pentose phosphate pathway. The body sees we have fructose 6-phosphate. We can use this for other stuff. Let's stop here. And it kind of does. And when I say it kind of does, this molecule stops at this step. What happens next is another molecule of zolulose 5-phosphate. So we get to ribulose and then convert it to a second zolulose. So a second one of these comes in. And now we don't have a sum of 10 anymore. We have 4 carbons and 5 carbons, so we have a total of 9. Using another transketolase, let me write that down, sorry. Transketolase will transfer, if I'm not mistaken, these two onto here to create a second fructose 6-phosphate and one G3P. Okay, and these can go right back into glycolysis. Okay, so this is fascinating because you can take fructose 6-phosphate and glyceraldehyde 3-phosphate, you can make them from the pentose phosphate pathway while also making NADPH. Okay, but that's not the only thing you can do with this. Okay, so for y'all who may or may not have taken a bio class, RNA is a ribonucleic acid. Ribonucleic acid. DNA is a deoxyribonucleic acid, which I'll just put up here, deoxy, and I'll just put a D like that, okay? The reason that it's called that specific name is that there's a ribose in it. Ribose, okay? So you guys are going to see in the next few questions is why I think the pentose phosphate pathway is so interesting, okay? So, oh yeah, regulation. We should talk about that first before we get into the interesting stuff, I guess. Okay. So, regulation. Before I get into regulation, I did forget to mention one other thing because it's not super important for the class, but it is something that is somewhat relevant. So, I guess it's not something I forgot to mention. It's just something I should mention more of. So NADPH, for y'all who don't know, well, should be everyone since I haven't explained it, but NADPH is used usually for fatty acid biosynthesis. There's a few other things it's used for, but that's its primary purpose. Okay. So G6P dehydrogenase, that's reaction 1. So in other words, are we going to go pentose phosphate pathway? Yes or no? Okay. It is inhibited by NADPH. It is also inhibited by intermediates in fatty acid biosynthesis. And I think that's why it's really important that I mentioned what I did just now. Okay, let's think about this in terms of why this is the case. NADPH is a big part of the reason we go through the pentose phosphate pathway. If we didn't make, or if we if we have enough NADPH, we don't need the oxidative branch, so why bother doing it, okay? Intermediates in the fatty acid biosynthesis pathway, exact same logic, right? If we've made some fatty acid using the NADPH, well, we don't need NADPH anymore, therefore we don't need the pentose phosphate pathway anymore, okay? All right, so now let me ask you guys, so I get why the oxidative branch is important. Why is the non-oxidative branch important? And that's kind of because we don't want to waste our sugar, is the short answer to that. Because ribulose 5-phosphate is cool and all, but the body doesn't really need it. So it converts it, several of it, into more usable forms. So I gave you guys the full oxidative plus non-oxidative, the entire pathway from start to finish, with 2, which is actually 3, ribulose 5-phosphates, because this is what the body does in order to not disrupt glycolysis. A single glucose, or I should say 3 glucose molecules go in, and you come out with 2 fructose 6-phosphates and 1 glyceraldehyde 3-phosphate, and you can just keep doing glycolysis. Nothing has changed. I mean, a couple of reactions aren't done, but effectively nothing has changed. Okay? But there's another reason, so the non-oxidative branch effectively converts ribulose into something usable. But there's another reason for it, and it comes down to what I was talking about with RNA and DNA up here. Ribonucleic acids, deoxyribonucleic acids, need ribose. And this is a great way to get ribose, okay? So the 2 main goals, oops, didn't mean to do that, apologies. 2 main goals of the pentose phosphate pathway. 2, and I'm going to put it in informal third. First, make NADPH. Second, make ribose 5-phosphate, which I'm going to abbreviate R5P. And then the unofficial third, make sure the leftovers, so the cellulose and the erythrose and the cetoheptulose and all that stuff, make sure the leftovers are usable, okay? Usable. So why do I make it seem like, so when I say that, it makes it seem like the non-oxidative branch isn't really helpful. But the reality is it's a lot more complicated. So these 4 cases that you see, 13 through 16, are different situations in which you can do different parts of the pentose phosphate pathway in order to get different things, okay? So looking at just number 1 for now, what happens in the pentose phosphate pathway when you need both ribose 5-phosphate and NADPH? It's very simple, okay? So let's agree on the fact that NADPH is only oxidative branch, okay? Therefore, you're going to say glucose in, oxidative branch, to get us a bunch of NADPH. And then when we're left with ribulose 5-phosphate, we use the isomerase reaction to get to R5P, and we stop. We don't need to go all the way to fructose or whatever. We want R5P, we're happy here, okay? So the pentose phosphate pathway can stop whenever it needs to stop to give the body whatever it needs in that situation. So in this case, we need NADPH and R5P. So the oxidative branch predominates, you get R5P right away, and you stop. You don't do the non-oxidative branch. You don't put stuff back in for glycolysis. You just stop, okay? All right. Now, what about if you need more of one of these than the other? So this is basically saying, let's just make as much of each as we can in number 13. Number 14 is saying, what if you don't really need NADPH that much, okay? And the answer is, you take fructose 6-phosphate and a glyceraldehyde 3-phosphate, and you do the non-oxidative branch, which I'm going to start abbreviating now, backwards to get to R5P, and once again, stop, okay? Because we don't need to go through the oxidative branch. We don't, like it's nice to have NADPH, but if we've inhibited the pentose phosphate pathway through having a high concentration of NADPH, so what we talked about in number 12 here, but we still need ribose. This is a great way to do that. Okay, right, and okay, right. So now other way around. What happens when more NADPH is required than excuse me, than R5P? Well, I mean, we can just do it, and it's more complicated than that. But effectively, what we could do is one of two things. Okay, so first, if you need, so I'm going to kind of make these one. So you put glucose in, and then you run it through the oxidative branch to get the NADPH you need, NADPH. Okay, and then you run it through the non-oxidative branch to get to F6P and G3P and all that. But let's say that's not enough NADPH. Okay, we want more of it. Or I should say this is the first situation. Let me put it that way. This is the first situation. If this is enough, then we stop right here, and these two just go on to do glycolysis. Okay. The second situation, and this is one that I think is fascinating because it's more likely, and it's really the point of this question, is, and I'm going to be writing down the formulas for this because otherwise it's too complicated. Okay. So first is we take 6 G6P and we send them through the oxidative branch. So at the end of it, we get 12 NADPH because we get 2 from each glucose. We get 6 CO2 because 1 from each glucose. And obviously 6 ribose 5-phosphate, right? Because we've only done the oxidative branch, right? Then we take this R5P, excuse me. So we take this R5P and using the non-oxidative branch, we're going to get 4 fructose 6-phosphate and 2 glyceraldehyde 3-phosphate. Okay? This is where things get interesting. And the reason I say that is because now we can use these two with gluconeogenesis to get 5 glucose 6-phosphate back, or glucose if we want to think of it as that. But effectively, the net reaction that we do is 1 glucose, or G6P really, plus 12 NADP plus gives us 12 NADPH and 6 CO2. So this is if we need to maximize our NADPH. And so we effectively, and this isn't the case really, but we're effectively converting 1 glucose completely into carbon dioxide. So we're oxidizing it fully. Now, obviously that's not what's really happening. We're taking 6 and oxidizing each of them partly. There we go, I apologize, I don't know why that happened. We're taking 6, oxidizing each of them partly, and then getting back 5. So the difference is 1. But that's how I think of it, is if we took 1 fully oxidized, all carbons, this is the reaction that we'd get. And this is if we really need NADPH, okay? But now what happens if you need NADPH and you need glucose, okay? But you don't really need R5P. Well, this is exactly like case 1 above. You take glucose or glucose 6-phosphate, whichever you prefer, feed it through the oxidative branch, get F6P and G3P, and these two will just do glycolysis as normal. Okay, this gives us NADPH. These two go through glycolysis and we get ADP out of it, or I'm sorry, ATP out of it. So we're happy. Okay. So the pentose phosphate pathway is awesome. It's really interesting. The oxidative branch can be done forward to give you NADPH and then lead into the non-oxidative branch, which can give you ribose 5-phosphate, which is needed for nucleic acids, or it can give you intermediates of glycolysis. You could use these same intermediates of glycolysis to go through the non-oxidative branch in reverse if you don't care about NADPH, or you could feed them through gluconeogenesis to make more glucose if you really want more NADPH and you really want to keep doing the pentose phosphate pathway. And so like I said, I love this pathway. I think it's fascinating. I love the way that it can kind of adapt to whatever the body needs so well. And so yeah, so this is one of my favorite chapters for that reason. I think I've gotten a little bit off topic talking about it, but if you have any questions, please feel free to ask them. Otherwise, next week we'll start talking about the the citric acid cycle, and then the following week will be the electron transport chain, and that's it for the content. So yeah, if you have any questions, always feel free to reach out or drop into peer tutoring. We're open 11 to 8 on Mondays through Thursdays, or 11 to 5 on Fridays and Saturdays. Yeah, thank you all so much for attending. I hope you have a great rest of your day and a great rest of your semester.